Osmotic pressure is the force driving water through cell membranes, and it's the mechanism behind one of creatine's most practically significant effects. Understanding osmotic pressure properly, not just the result, but the physics, clarifies why creatine's water retention is fundamentally different from the fluid retention associated with high sodium intake or inflammatory swelling. Osmotic pressure arises at a semi-permeable membrane when the solute concentrations on either side are unequal. Water moves from the low concentration side to the high concentration side to reduce the concentration difference. The force of this movement, the osmotic pressure, is proportional to the solute concentration difference. Inside a muscle cell, the primary osmotic solutes are potassium, proteins, amino acids, and E in creatine-loaded muscles, creatine and phosphocreatine. When you supplement creatine and it accumulates inside muscle cells, the intracellular solute concentration rises. The extracellular fluid surrounding the cell, the interstitial fluid, has a fixed osmotic pressure determined primarily by sodium chloride concentration. The concentration difference across the cell membrane drives water in from the interstitial space. This water influx is passive, proportional to the creatine accumulation, and stops when equilibrium is reached, when the cell's internal osmotic pressure matches the external. The resulting cell is physically larger, has higher internal hydrostatic pressure, and has a different mechanical property profile than the same cell without extra creatine. What's different about sodium-driven fluid retention? When you consume a large amount of sodium, it equilibrates between blood plasma and interstitial fluid. But sodium doesn't cross into cells in significant amounts. The excess sodium in interstitial fluid draws water from blood into the interstitial space, the tissue between cells, not inside them. This produces subcutaneous swelling, puffy face, swollen ankles, softer-looking muscles. This is the kind of fluid retention that makes athletes look less defined before a competition. Creatine's osmotic effect is in the opposite cellular compartment. Inside, not outside. The practical consequence for anyone making physique decisions. The water that creatine adds makes muscles look bigger from the inside out. The water that excess sodium adds distributes in the interstitial space, blurring muscle definition. These are not just quantitatively different, they're qualitatively different in visual and functional effect. Inflammatory fluid retention. The swelling after injury or during illness is also primarily interstitial, not intracellular. When tissue is damaged, the capillaries become more permeable and leak fluid into the interstitial space. Again, this is the opposite compartment from creatine's water. The three types of fluid retention, osmotic creatine water, sodium interstitial water, and inflammatory leak, are entirely distinct in mechanism, location, and physiological consequence. Treating creatine's water as the same category as these others is the error that drives the puffiness concern. Understanding osmotic pressure makes clear why creatine's water is an asset, not a liability. The physics of osmotic pressure also explains why body composition measurements can be misleading in creatine supplementing individuals who are also cutting body fat. Standard body fat percentage methods, bioelectrical impedance, DEXA scan, hydrostatic weighing, all have assumptions about body water distribution built into their calculations. Bioelectrical impedance, the most common method, estimates body fat percentage by measuring how electrical current moves through the body. The calculation assumes a standard relationship between lean mass and body water content. Creatine's intracellular water retention violates this assumption. The lean mass of a creatine supplementing individual contains more water per kilogram than standard tables assume. The result is that bioelectrical impedance typically underestimates body fat percentage in creatine users, or conversely, overestimates lean mass percentage. DEXA scan is less sensitive to creatine's water distribution because it measures bone mineral density and soft tissue mass directly, rather than inferring composition from electrical properties. But even DEXA reads lean mass as the sum of all non-fat tissue including water, so the extra intramuscular water from creatine adds to the measured lean mass. This means that when you start creatine, your DEXA-measured lean mass will increase by more than your actual muscle protein gain in the first month. And when you stop, your measured lean mass will decrease by more than your actual muscle protein loss.